Hello students, welcome back to the biology class. So in the last class we started the concept of metabolism. We understood that metabolism term was coined by Shawn in 1839, and metabolism can easily be defined as all the chemical reactions that go inside a living system. And we all know that biomolecules are formed during the metabolism, biomolecules are utilized during the metabolism, and they are even undergoing transformation from one form into the other form during the process of metabolism. So this rate of biomolecule formation and their utilization and transformation is called as the turnover rate or turnover. Now metabolic pathway is not a single step process, it's a multi-step process. In examples you'll be learning when we will go to the plant physiology unit and the human physiology unit. So there are many reactions which are going on in the stepwise manner, even inside a prokaryotic cell, even inside a eukaryotic cell. And these metabolic pathways can either be linear pathways or circular. So I told you the example that glycolysis, which is the first step of respiration in every type of respiration, is glycolysis. It's a linear multi-step metabolic pathway. And in case of the eukaryotic organism, because mitochondria is there, so after glycolysis the next step is a circular pathway which is called as the TCA pathway. And all these multi-step metabolic pathways are linked with each other, and then they are controlled by the enzymes. Now metabolic pathways may lead to the formation of complex substances or breakdown of these complex substances. So accordingly, two types of metabolic pathways are there: anabolism and catabolism. Anabolism is constructive pathway, catabolism is the destructive pathway. Anabolism performs build-up reactions and catabolism performs the breakdown reactions. Anabolism it produces the complex substances from the simple substances. So you can see from simple substances a complex one is formed, and in the case of catabolism, simpler substances are formed from the complex substances. In this case, the energy, in the case of the anabolism, energy is changed into the potential energy, or you can say there is storage of energy. In this case, in the case of catabolism, energy is released, so potential energy is converted into the kinetic energy, and this is required for growth as well as for storage as well as for biosynthesis. And catabolism is required for, basically is performed for the biosynthetic pathway. So these are the major differences, and if you wish to learn, easiest example, respiration is catabolism, photosynthesis is anabolism. Or respiration is catabolism, fat build up in the body or protein build up in the body, or muscle build up in the body. These are examples of the anabolism. Remember, every living organism has to be always performing functions, then only it can remain in the living state. So that is only possible in the non-equilibrium state. And we learned one more term in the last class, that was the amphibolic pathway, that the compounds from the anabolism or the intermediate products of the anabolism. Why intermediate? Because there are many steps. So any one step product may go from one reaction to the other reaction, or from one metabolic pathway to the next metabolic pathway. So things can move from one pathway to the next pathway, so it can easily be called as amphibolic pathway. We started the topic of enzyme in the last class, and we learned that enzymes, the word enzyme was coined by Willy Kuhne, but this name became popularized because of Buckner, because he performed an excellent experiment, and his experiment was regarding sugar converting into alcohol. By that time everybody knew that yeast is able to perform conversion of sugar or fermentation of sugar into alcohol, but what Buckner did was he killed the yeast and took the extract out of yeast, and then he used the extract of the yeast in the sugar solution, and to his surprise, sugar again turned into alcohol without the presence of the living yeast. So it became clear it is not the living yeast which is responsible for fermentation, it is something inside it. So from there he started calling it as the zymase. So zymase was responsible for the sugar to change into alcohol, and this is how enzymes became very peculiar or familiar. Now the next thing, why was it called zymases? Zyme word is used for the yeast. So because this enzyme came from yeast, that's why it is called as zymase. And from the same enzyme, A means in, so it is obtained from the enzyme. It is obtained from the zyme, or you can say yeast. That's why the first enzyme was extracted from yeast. That's why the name became enzyme also. So enzymes are the biocatalysts. Catalysts are something which are able to change the rate of reaction, and because they are working inside the living system, so they are called as biocatalysts. Then they were discovered by Berzelius in 1853. So he knew that something is there, but could never properly make it people believe. And then remember, enzymes change the rate of reaction in every small, they are required in very small amount, and they are not consumed or changed during the course of reaction. And enzymes can have a positive effect on the speed of the reaction, or it can have a negative effect on the speed or the rate of reaction. And remember, maximum enzymes are protein. But there are few enzymes, which I told in the last class also, few enzymes which are made up of RNA. And the enzymes need an active site to function. An active site is created by the tertiary structure. That's why enzymes can never be fibrous proteins. They can be only globular protein, because they need the tertiary structure. Now Sumner performed the purification and crystallization of urease enzyme. He also stated that the enzymes are made up of protein. And finally, this was confirmed, that enzymes are made up of protein was confirmed by Northrop in 1936. Then we learned this. We learned what are bioreactor. This was the last statement in the class. Now let's understand next point now. Enzymes can be of two types. So some are called as housekeeping enzymes.